Hi guys!
Hola!
It's time for a lab on osmosis. This is osmosis in the plant cells.
Okay, so you guys just finished the PowerPoint where we kind of talked about the difference between osmosis and diffusion. So now you are going to actually put that knowledge to work. So we have some background information here on the first screen. So if you want to go ahead and pause the video so that you can read through that, go for it. We are not going to read it to you.
Nope. Okay, so the problem statement or textbook question for this lab is how does salt water affect the cell membrane and cytoplasm of LLDL leaf cells?
Okay, so then you have your materials. There's not anything in this material list that you haven't seen before. The only thing new that you haven't used previously is a salt solution, but that's basically like salt water. So if you've ever gargled with salt water before,
Ew, I hate that.
That's what we're making.
Yeah. Okay, so your procedure, of course, we need you to wash and dry a glass slide and cover slip, and then you're going to make your slide. You're going to make your slide like you've done before, a drop of water, one single leaf, and then add your cover slip, and you know how to do that at the angle.
So then you're going to go ahead and take a look at your leaf underneath your microscope. Make a detailed drawing of a few of the cells so you can make sure you document the shape of them. Okay, remember to start on low power, but then you want to work your way up to high power. Don't draw the entire field of view. What that means is we don't want to see everything that you can see. Just draw us a few cells, so 2 or 3.
Yep, that works for us. Okay, continuing on. And then you're going to do what you did with when you dyed your cheek cells. You're going to take one large drop of salt solution at the edge of a slide. And then you're going to put your paper towel on the other side and draw that salt water underneath. So you're looking at the cells with normal water, and then you're going to add your salt solution. One big drop or a couple small drops. Make sure that salt solution is touching that cover slip, and then it will draw it through like you did before.
And the salt solution should already be prepared for you.
Yeah.
So it'll be in a beaker with a label on it, so you can get your salt solution from there. So once you've put your salt solution next to your cover slip, and let it kind of absorb under your cover slip, and you are ready to put it back under the microscope. Once again, start on low power, work your way up to high power, and you are going to take a look at how the cells have changed. Look specifically at their shape to see if you can notice anything different. So you're going to do a drawing again. Make sure you label your drawing. Don't draw the whole field of view, just a couple cells, 2 or 3, so we can get an idea of the shape and what they looked like at the beginning versus what they look like now.
Yep, and we want you to use color pencil with this thing so you can really communicate what's going on there. Okay, so what you're kind of looking at, this is an animation, or just a drawing, it's not a real life view. So what you should be looking for is how it's full in the beginning, and then afterwards it kind of got shrunken in a little bit.
So you should definitely be able to see a difference in shape and probably size as well. So as you're kind of noticing these differences, start thinking about what could have caused the differences. Think about what you changed and why would that change have created this after effect.
Okay guys, so it looks like that is the end of our show.
Make sure that you answer any questions that follow the end of your lab that you have. So you should have a lab sheet with you. Use complete sentences. Make sure that you are using specific words, not things like it or them. Okay, give us details.
Nouns.
Nouns. And if you have any questions, come see us.
Yeah, it's really tricky why the cells collapse at the end. So make sure you ask for help if you don't understand this concept. We'll be glad to explain it to you, but we don't want to tell you now because it kind of ruins it all.
All right, see you later.